All right. Good afternoon, everyone. Good everyone, wherever we are joining us from. I hope I'm audible. So you can do it to make use of the check mark. So I'll just do that so that whenever I'm in call, you guys will be able to. If I do have any questions to ask you, you will be able to, make use of the check mark or make use of the thumbs up to just help me in confirming that what I'm saying, you are getting what I'm saying. So you can either use the thumbs up in the check using the reaction button in the thumbs up, and also you can use the raise hand button if you have any questions you want to ask. So about the section. All right. So let me start by sharing my screen and hope you can see my screen and at the same time you can see my face. So can we see my screen and we can see, okay, all right. So we can also see me on the call. Is my face visible? Okay, all right. So, and you can also see my slide. So I'm currently on my slide. Maybe I should, okay. My slide. So, good day, everyone. It's nice to welcome everyone here. And I can see a lot of people joining the call. Let me quickly switch back and check. I can see Ayomide, I can see Joy, I can see Constance, I can see Wilson, IK, and some other people joining the call. I think I can see some familiar names here, though I'm not sure I'll be able to picture a face to that. So, good afternoon, everyone. Again, it's nice to see you guys joining the call on bioinformatics, how to do some bioinformatics analysis, using our programming, and it's a pleasure to welcome you to Data Lab to see what some of the things that we are doing at Data Lab when it comes to training. All right. My name is Daniel Adede, and I'm a bioinformatics and health data analyst at Data Lab. Majorly, I focus in training student interns around how to analyze healthcare data, doing end-to-end healthcare data on the aspect of using Python and then using some other programming languages, and also the bioinformatics aspect, how to analyze bioinformatics aspect. And I'll be sharing some of what we do teach at Data Lab around bioinformatics in this particular course. All right, if at any point in time you can't really hear me, or if I'm not audible enough, if my network is breaking, please do wait to let me know. So today we would, for some of us that are just hearing the word bioinformatics for the first time, so I want to quickly uh, talk what bioinformatics is. I can notice that some people are here. This is your first time you are joining the call. This is your first time you are informatics, right? So, and I would like to explain that. So what bioinformatics is, I would have loved to ask us questions, but we have a lot to cover today, and I will not be able to do that at the moment. So what is bioinformatics? So bioinformatics, basically, just think of it as using computer technology to solve what biological problem using what biological data. So that's basically bioinformatics. So the art of digitizing what life science. So that's bioinformatics, and it will feel responsible for storing, analyzing, and interpreting big data generated by life science studies or acquired in a clinical setting. I'm very sure that most of us here on the call, if this is our first time hearing about bioinformatics, I'm very sure that we are familiar with what DNA is, even though maybe we are not coming from a life science background, but I'm very sure that we've heard about DNA before, what a DNA is and what protein is. So all these are data that are being generated by life science studies, life science research studies, and we make use of our field of bioinformatics to analyze all this data, to study data, to analyze data, and then to interpret this big data. Bioinformatics, just like any other field out there, is driven by experts from several disciplines. Basically, for bioinformatics, you would always see a biologist, you will see computer scientists, mathematicians, statistician, and physicists. Physicists coming from the angle of you sequencing your DNA molecule, and biologists basically, you know that you have to interpret it's a biology data, it's a molecular biology data, and then computer science developing the algorithm, the mathematics, that's the idea behind how this algorithm has been developed, and the statisticians using statistical models to analyze the data. That's pretty short about what bioinformatics is. So we do have some applications of bioinformatics. So one of which is drug discovery. So one of which is drug discovery. Bioinformatics as aid in uh, so using a structure-based design with the knowledge of bioinformatics, scientists can come up with effective drugs. So I hope I'm very audible. Please let me to confirm that. Can't say the
Yes.
right. Uh, so with the knowledge of bioinformatics, scientists can come up with effective drugs for various illness and acute diseases using some of these following approaches that I will be explaining. The first thing is target identification. So with bioinformatics, we can quickly identify potential drug targets via doing genomics and proteomics analysis. And I will not want to bore you with all what genomics and proteomics analysis is. I just want you to know that whenever a scientist or drug researchers want to develop a candidate drug, they need to see the target point of that drug. So with the knowledge of bioinformatics, we can easily and quickly identify potential drug targets using some of omics analysis, basically genomics and proteomics. And after that, these computational drug scientists will then go further into designing a drug molecule that will fit into the target site, that will fit into the identified target site. So that's another angle, so target identification, then drug design. And after that, so you want to see what the interactions between these drugs and what the target in question. So you want to see if there's going to be a lock and key a relationship between the target and what the drug molecules. You want to see if the drugs will be able to bind effectively to the target, not only binding, also able to, there are some keys that would fit into a lock, but they will not open a lock. But you want to be very sure that these same drugs will fit into the target and will be able to, open and be able to unlock the target, will be able to cure disease via unlocking the target. So these are some aspects of drug discovery process that bioinformatics are really very important, that you can leverage bioinformatics to solve some of these problems. And then let me show us something here, a conventional way of drug discovery. So you can see from here that we do have the target-based approach. Is either from conventional ways, either you have what target-based approach or you have what cell-based approach to to develop drug or to uh, discover a candidate drug molecule. So you either have a target-based approach. The target means that you already know the target point you are looking for at the molecular level. So you are looking either at the proteomics level, either at the transcriptomics level or genomics level or some other omics modality. You are looking at it from that target point, or you want to target at the cellular levels, that cell-based approach. You want to target at the cellular level. So in conventional way of drug discovery, it goes for a very, very long time to bring a candidate drug molecule to the market. So you go through the process of what uh, identifying the target, maybe screening over thousands of molecules just to identify the target. And this always involve partnerships between universities and industries just seek for financial support, especially the big pharma company would always fund the startup that are just coming up and developing candidate drugs. And you go through the screening process, then identifying the lead, and then doing the optimal for a disease. Then after that, through a lot of IRB process, ethical review process, then do phase two, phase three, clinical trial phase, then some government policies around maybe EMA and then FDA, just to see, validate if this drug molecule would really work for the populace, and then begin the manufacturing and then to the market. So this takes a very, very a serious process, and then at the same time, some of these candidate drugs may fail. Majorly, most of them do fails in reality before getting to the manufacturing phase and then to the market. So this is some of the reason why you will see that most drugs are very expensive, majorly because they are trying to account for some of the loss faced why some other candidate drugs are that fails during the process of during the pipeline development of the drugs. So this is one of the very like tedious way of developing candidate drugs. But with the help of bioinformatics, you do not really need much of fundings to bring the candidate drugs to the market. You just need to screen molecules using computational approach. Then you synthesize the lead candidate that you have at the end of the day. Then you send to preclinical phase one, phase two, phase three. Now, I'm still going to explain more about this using the vaccine approach to explain how bioinformatics have been of help when it comes to the synthesis of drugs and biologics. So another subfield of bioinformatics is what immunoinformatics. Bioinformatics is a very broad field, like I said. It brings biology, computer science, then mathematics, and some other fields like that uh, on board. And then this bioinformatics is a subfield, and you do have this bioinformatics has a lot of subfield. We have immunoinformatics, we have chemi informatics. That's the aspect of drug discovery. So that shows that bioinformatics is very broad. You just need to focus on a particular niche, what you are really interested in doing with bioinformatics. But this is approach for vaccine development. Vaccine is biologics, right? Not drug. Vaccine is biologics, not drug. I think FDA also have some regulatory aspects that are in charge of overseeing to the biologics molecule. I've forgotten the name for now, and also for what drug molecule as well. So, but for vaccine development, you have to go through some process like trying to isolate the viral organism and trying to maybe knock down the virulent genes and some other steps like that. But with the help of bioinformatics, we can use reverse vaccinology approach to pick the candidate, something that we have seen maybe from literatures that would help in developing a candidate vaccine, either selecting the viral genome, you selecting the viral genome, looking at the viral genomes, especially. I will still explain more around this in my next slide. That is if you are designing the vaccine for if you are designing the vaccine molecule for infectious disease, viral, bacterial genome, any infectious disease organism, just need to look at the genome, the sequence of the genome. You check it out in the database, and if you are designing the vaccine for maybe cancer or some other genetic diseases, so you want to pick the host genome, but I think that is still the nascent phase anyway, because you are designing vaccine for the host. That means there's possibility that the generated antibodies will also be attacking the host, but there's still a way to maneuver that using some techniques to target that particular genetic cell, aberrant genetic cells. So now, after identifying that, we need to we need to get the genome of the viral organism. I'm going to be using viral at this point in time. You just After that you select vaccine target based on what bioinformatics analysis. We do a lot of analysis to just select our vaccine target. So after that, then you send for synthesis and you clone a vaccine target. You express and amplify this vaccine target, and then you immunize mice with this vaccine target. You see the level of antiserum activities in this vaccine target. And if this is not okay, then you can go back to the step of you optimizing this again. But in the case whereby you are using conventional approach to design vaccine molecule, it's going to take you a lot of effort, a lot of finance to be able to do that again. But with the help of bioinformatics, you see the effect of what the vaccine already. You surely know where to to optimize the vaccine candidate to see if you pass it through this process again, you would have a good immune antiviral activities at the end of the day. So and then after that, if you are very sure that this is okay at the preclinical trial phase, then you go to the clinical trial phase. I'm still going to explain more about this in this next slide. If we just picture what happened during the COVID-19 era, like the pandemic era rather, so you can see that COVID-19 started December 2019, right? And at the end of 2020, at the end of 2020 to 2021, we already have series of approved vaccines. We have approved vaccine at the end of 2021. So this is as a result of bioinformatics. Hadn't been, we are still using the traditional approach of vaccine development. That would take years. That would take 10, 15 years to do that. But the with the help of bioinformatics approach, we are able to bring a candidate vaccine to market in just 10 to 1.5 years, 10 months to 1.5 years times. So although this requires fastness on things, just because because we are in a pandemic era, so we need to fasten some process. But with vaccine molecule in the market within 10 to 1.5 years. So this is the approach. You design and explorative preclinical studies. You pre-existing information about SARS-CoV-2 and MERS-CoV-2 to design the drugs, and then you process it, you submit to R&D, then phase one, phase two. These are overlapping clinical trial phase, which is not the best to do things, but because it's demanding. We are in a demanding situation at that point in time. So that's why the trial phase is overlapping, and then to the regulatory phase, then to the market. But with just know that bioinformatics aids in early drug and vaccine development. So now in the agricultural space, how do you think vaccine will be useful? So I've talked about drugs and I've talked about vaccine development. So and I'm going to open floor for us at the end of my first presentation before moving into the hands-on section. If we do have any question to ask before moving into the answers, then I will, and you can put that questions in the chat just to be sure that you didn't forget those questions before my presentations. And so in agricultural biotech, you know how biotechnology has been very useful in the agricultural space. You know, it increased crop yield. So using genetic engineering mechanism now to introduce some even under poor soil or limited water condition. And this is crucial for meeting the food demands of the growing populations. And biotechnology has been helping in that regard. It improves crop yields and then disease and pest resistance. Uh, Introducing works on around biotechnology space, and this reduces crop losses and ensures stable food production and also sustainable farming. Very sure that must be hearing about GMOs. GMO genetically modified organisms require fewer chemical inputs, minimize the environmental impact of all these chemicals, and this leads to healthier soils, cleaner water, and reduced greenhouse gas emission, supporting a sustainable agricultural practice. So, how does bioinformatics first thing is you sequencing the genome of that crop, then you identifying the genes for desirable traits you are looking for. Maybe you want to make the crops to be resistant to disease or pest. So you know the candidate trait, the genes that you'll be targeting. And let's say you want to increase the crop yield, you know those genes that you'll be targeting. And after that, you have to do a lot of analysis around the genomic data to enhance plant breeding programs. And then you use bioinformatics to forecast. You use bioinformatics to forecast the outcome of uh, genetics modification, so guiding experimental design. To ensuring food security, this approach would ensure food security, promoting environmental sustainability and drive continuous innovations and crop science. So this is how bioinformatics can help to in the agricultural biotech space. So now to genetic epidemiology, so which is one of the step approach of what we will be doing in the hands-on section today. So you want to genetic epidemiology basically means that you want to identify patterns and cause of outbreak, especially in infectious disease, by tracking what the genetic mutations. You want to predict the chance of susceptibility to disease through genetic data analysis for better preventions and treatment. You know, with the help of uh, genomes, you'll be able to see if this person is predisposed to cancer or some other genetic disease via looking at the genetic makeup data, and that's genetic epidemiology. And also in population studies, you want to see why some ancestries, like African ancestries, are more predisposed to breast cancer. I'm just using, for example, compared to European or American ancestry. So, for example, individuals of Eastern European Jewish descent have high rate of breast and ovarian cancer when compared with some other ancestry. So, you want to see in the genetic makeup that why is it that some set of populations are more predisposed to a particular disease when compared with others. So this is genetic epidemiology, and with bioinformatics, you can picture all this, you can see all this, and what is which genetic as which genome is or gene or a variant in the gene that is causing this predisposition to this genetic disease. So this is just what happened, especially in genetic epidemiology, feed genetic epidemiology, and using, collecting epidemiological data, then clinical data and population data, genomic data as well. So talking about the common epidemiological data that we will be collecting, like the contact tracing, the travel histories, the patient demographics and some other informations. You cannot talk about genetic epidemiology without talking about all this epidemiology data and also clinical data and population data. So combining all these data together to gain insights, I think with the help of genetic epidemiology, we were able to trace down uh, COVID to what China, Wuhan, China. So with the help of genetic epidemiology, we were able to do that, and this is possible with bioinformatics, using bioinformatics approach, analyzing all this data, both the genomic data, epidemiology data, clinical data, and population data. So that is that. Now to just that is just a brief introduction of what bioinformatics is and the applications of bioinformatics. How bioinformatics has been helpful in the space, in every space, every field in the life in the earth space. So now to the phylogeny. Phylogeny is what we'll be talking more about in this section, in the hands-on sections. I just want to do a quick introduction in two slides what phylogeny is. So phylogenetic tree is also known as phylogeny, and it's a diagram that depicts the line of evolutionary descent of different species, organism origin from a common ancestors. So, and phylogeny attempt to reconstruct evolutionary ancestors. Like you want to see the evolutionary relationship between man and, let's say, chimpanzee or monkey. So you want to see if there's evolutionary relationship between, estimate time of divergence from ancestors. Who, like what time does this divergence really occur between these different organisms or between these different genes. When we talk about phylogeny, we are not just looking at it at the gene level, we are not looking at it at the species level, we are looking at it at the organism level, we are also looking at it at the genetic level, which we will be doing today. So we do have different, just out of the slide a bit, we do have different phylogeny, but what we will be doing hands-on today is going to be what molecular phylogeny, but we do have some other phylogeny like molecular rather we do have morphology phylogeny, like what you are seeing physically. For example, if you should look at chimpanzee, you look at some other apes, you'll see that uh, very much pretty alike, right? So that morphology are using the what you can see to see if there's evolutionary relationship between these organisms in questions or these species in questions. And that morphology phylogeny. But molecular phylogeny, you are looking at it at the genomes level. You are looking at it at the gene level, the molecular data level. So that's molecular phylogeny. So this is what we'll be doing today. We will be looking at the relationship between different organisms at the molecular phylogeny level. And so what are the applications of some of these phylogeny? Um, Phylogeny can be used to solve a number of interesting problems like forensics. So you want to be sure that if we want to be sure if there is relationship, like if you can trace down a particular event to a locations where that happens or to a particular person. So that you can use phylogeny to do that. That's forensics. And you want to see how HIV virus mutates rapidly. So how does it mutate rapidly? You can use phylogeny to do that. And another thing is, just before moving to the next applications of phylogeny, I'm very sure that most of us, they are pretty much conversant with different variants of SARS-CoV causing what COVID-19, right? So we are very conversant with like different variants, like we have the alpha variants, beta, gamma, omicron variants, and some other variants like that. And it as a result of phylogeny and doing some nesting analysis that we were able to identify that this omicron variant is more pathogenic when compared with the alpha variant we have at the first instance. So the phylogeny would help to see the different variants that we have, and also with some other analysis to be able to identify if this variant will be more pathogenic with these previous variants that we have. So that's one aspect. And you also want to predict evolutionary relationship between not only virus, any organism mentioned, you want to predict evolutionary relationship between many different organisms. So, like influenza virus, we do have different variants of influenza virus. We have H5N1, H7N1. Just mentioned it's using the H and N for the variants. So, we can really do evolutionary relationship between these different influenza viruses. And also, with the help of phylogeny, we can predict functional relationship between especially uncharacterized genes. Maybe some genes are not known, so and we have genes that are known in some other organisms. Some genes in human are not known, and we have genes that are known in, let's say, chimpanzee, monkey, some other apes. So we can just pick that uncharacterized genes and then try to compare with known genes in these different like chimpanzee and others. We can just see if there is going to be a kind of evolutionary relationship between them, and from there we can really predict the functions. But that is not to conclude that that gene is actually performing this function, because genes can do have the same molecular information, but their functions may be different. So we still have some other analysis to do. So that's autologous detections of the genes, like you plotting a phyllo tree and then trying to see if the genes are performing the same functions. So that's another aspect of phylogeny. And phylogeny can also help in drug discovery, vaccine development. I'll just have this around just to see if, like, uh, I've been talking about different ancestries. Talk about African ancestry, we have the European, Asian, and then the American ancestries, right? So you would want to, let's say you are developing vaccine molecule and drug molecule, and it has to do with the genetic makeup of human. So you need to try to compare, and you are developing it at a global scale. You are developing it globally. You want that drug to be very effective for all these ancestries. So that means you can do well to plot phylogenetic relationship, and you want to see if there is no diverse time, like the timing for evolutionary relationship is not that high for that particular target, target genes or target protein is not that high. So you want to see the relationship between these different ancestors at the phylogenetic level. So that's on the aspect of drug discovery and vaccine development. So I'll just shut up at this point, and if we have any questions to ask before moving to the answers section. Do you have any questions, comments before moving to the answer? You can do it to drop your questions in the chat if you do have one. I will just wait for 30 seconds more before switching to the answer section. And I would like to ask, are we done installing the R and R Studio that was sent to us? Now, I hope I'm audible. Okay. Faith is asking, what I've shared regular occurrence. I don't know if you can turn your questions more so that I can get the aspects you want me to answer. If you can do it on mute. I think the question you ask here is very broad.
Hello, can I talk now?
Yeah, I can hear you.
All right.
Audible.
I just want to understand, being new to the field, right? I want to be sure that, these applications of genetic epidemiology, the phylogenics applications of um, bioinformatics are regular projects one will be able to find to develop one's skills in the field.
Okay,
Do
yeah,
you
yeah.
understand
Okay.
me now?
Yeah. Thank you for
Is
that.
it easy to find projects that one will be applying the knowledge we get? Since you have said most of the things we'll be talking about have to do with genetic epidemiology and phylogeny. Will it
Yeah,
be easy? Is it easy to find projects that will be using these skills in?
yeah, very much easy because we do have a lot of data out there, biological data, and some of these data we will be using it during the hands-on section. But when you come to like practicing it in the real life scenarios, that means you are part of some governance. But some of these are publicly available as well. But when it comes to having each, maybe you are talking about viral and the infrastructure as well, because the infrastructure may not be in place, except if you are partnering or working with an organization where you can really have access to all these data, like real-time data, I mean. So I think that's another thing. But if you want to work via practicing, there are a lot of data out there to do phylogenetic analysis. So which I'm still going to be doing using some of the publicly available data out there to do the practice today with you guys. Okay, does that answer your question?
Yes. Thank you.
Okay, all right. So the recordings, and you will surely see the recordings very soon in the YouTube channel. We do post our recordings in the YouTube channels. So that will be sharing the YouTube channel. So yeah, we are pretty very sure that this you cannot, if this is your first time coming into the bioinformatics space, it's not going to be easy for you to get everything within 2 hour sections, right? So I'm still going to be explaining more about that later on at the end of the call, but we just want you to follow through. I'm pretty sure if anybody should ask you what bioinformatics is right now, you should be able to give a brief explanation of what bioinformatics is and some of the applications of bioinformatics, what phylogeny is. And I'm going to be, at the end of the call, I'm still going to talk more about how to go further into learning more and the likes. Okay, so if there's no other questions, I think I can move forward to the answer section. Let me start by sharing my down. Okay, so I'm just so my Arc Studio. I'll open my Arc Studio. And I want you guys to do the same thing. And I'm going to be sharing a file with us in the chat so that we can follow along. I'm sharing a file with us in the chat. Can do it to download that and let me know if you are done doing that. So if we are done downloading the file, please let me know. Okay. I need the response. Okay. I see 2 responses. I need two to three more responses so that we can move forward. Yeah, I can see 3 responses now. Then I'll just switch to 1250, then we can move further. Okay. Need one more. Okay, all right. I think we can start. After downloading the file, I'm pretty sure that that file will still be in your download folder, right? What you need to do is after opening your what's it called, your Arc Studio. So I want you to go to, and I'm going to be explaining a lot of things there, and I want you to just follow me too. So you come down to this place to files, then you said open file. And then for me, I think I did this before. So I'm directed to this place. For you, it may be on the desktop. So what you need to do is maybe go to the download folder. Go to the download folder and then, click on that. Kotu markdown file. You'll be seeing it, and I'm still going to explain more about that after loading that. So for me, that file is not in this desktop, in this download folder. So I need to move towards that bioinformatics analysis folder, and then you'll see something like this. This is the QMD file. So this is the QMD file. Just click on it and then what? Open. So I have it here already. That's why it is not reopening. Hadn't been I close this. Let me repeat myself. Let me close this. Before going further, I will still explain more about that, but I just want you guys to load the file so that I will not keep stopping us every time. So I'll click this and then open. You can see that we have the file here, right? I'm not interested in this untitled. This is just by default. I'll delete it. This is the bioinformatics QMD. So I want to see if we are done loading this QMD file to the folder, to the environment rather. Yeah, I sent the QMD file to the chat. You mean the Arc Studio that I'm using is different from yours? Not not sure we are getting this one from, but so yes, I will share the link now, so that you can have it as well. Okay, I think you can see it now. Send it to the chat. So I've already sent that. Can you see that now? Okay. So I think did you follow the step that was sent to you via email? Like you download your R first, then followed by you downloading the Arc Studio, the instruction that was sent to you. I'm not sure what you are saying about RQ. Uh, Somebody said the Arc Studio you are using is different from mine. Can you come down to this search just to be sure that you downloaded the Arc Studio? You see Arc Studio, then you click open. You click open Arc Studio, not R. It's saying our graphical user interface. Right. So what I'll do is don't want this to be. Just want the. You are done downloading the Arc Studio as well. Have you downloaded the Arc Studio? Because it's one thing to download the Arc, it's another thing to download the Arc Studio. Do we have somebody that followed the instruction? I think I'm going to. I'm coming. I think this is the instructions, right? Did you follow this instruction to download Arc Studio? Because this is the instruction that was sent to you. This is Arc Studio, and then if you're a Windows user, you download Arc Studio Desktop for Windows. So, and if you just want to use this, you can use this for Windows. If you're a Mac user, you use this. Did we follow that? There's one thing to download Arc. So that's pretty what you guys did. I'm not sure we followed the instruction to download Arc Studio. Okay, so now I think I figure where the problem is coming from. Wilson said he has downloaded the Arc Studio, and it's you that. Wilson, please can you respond so that we can move forward?
Yes, so what's the question, please? Sorry.
Or if you guys, he said.
Please, what's the question? I've downloaded our studio. I've even opened the QMD file.
Yeah, I think we are on the same page, right? You can see what I have here.
Yes, it's the same thing.
Okay.
I wanted to do it. Okay, sorry.
Okay, I think that's what I want to hear. Thank you so much. For practicing sake, if you are yet to download that, I just want to use 2 minutes to quickly explain. Hopefully you can follow that. But because some of us, our network may not be very stable, that is not encouraging to do most of the time. It's good for you to go back and download Dark Studio to be able to, have the same fleshing interaction with this interface in your system rather than on the cloud. But because some of us are not, you need to create an account. I'm going to send this to us. Hopefully we'll be able to follow on time. You need to create an account, and then you come after creating the account, you say you want to new project. For me, I will just say new project, Arc Studio project. I just want you to follow it like that. So after creating your account, you have something like that, new project. Then so you have something like this loading popping up that your login has been successful. So something like this. So and then for you to upload the QMD files that I sent to you, I'll just make this very fast because we do also have a lot to cover. God, still loading, all right? So now advise able to use this because it's network. So what if you are doing this in an environment where the network is not stable and you really want to carry out some analysis? So still loading, think. So you have something like this, right? When you open this is in the browser. You just come here, you said open files, the same way we did it. Open files, I think I want to open file from the system. No, this is not the way to do it here. So I can't open file like this within this. Come to this upload file, and then you choose file from your system. So mine is going to be desktop, bioinformatics analysis, and then towards bioinformatics, this QMD file. For yours is going to be on the download folder, I'm pretty sure, or else you reset your downloading to a particular folder. So this is bioinformatics, then open, and then OK. So now you will see this bioinformatics QMD folder in this plot page. So I was still going to explain more about that, and then you open it. It's going to open here. It's going to open here, and you have something like this. I think it's still saying app down is not installed. Basically, that's why it's not displaying. Yours will stay the same. You just click install. That would work, but because of our time, that will be installing. So I'll come back to that. But because of our time, let me explain more about this. So if you follow through the R and Arc Studio instruction sent to us, you should have something similar like this. All right, so now you have the QMD file again. So if you have something like this, this is R Studio. The programming language is what? R. The programming language is R. This is R Studio. But because it's our environment is not intuitive enough, it's not interactive. So it's there's a particular, let me say, Arc Studio. That's what they are called. They are now called posits, right? So they develop the Arc Studio. They are now called posits. They develop the Arc Studio as a user interface, but running the engine that is running this Arc Studio is R itself, the R programming. So that's why I said the first thing you need to download is what? The Arc, then followed by the Arc Studio. The recording will be provided. You surely see the recording on our YouTube channel. So the Arc Studio is just like interface. At the backend is the Arc engine working. And now this is the Arc Studio. Now you have Arc Studio has 4 pane. That's 4 pane. So this is the first one, this is the second one, this is the third one, and this is the fourth one. This is first one, second one, third one, fourth one. You can do it to interchange it if you are not interested in it. But this is the default format that Arc Studio is going to come whenever you open it. But you can do it to interchange it. If you are interested in this, coming to this, depending on how you like it. But this is the default format, and which most people do use writing your code. So now the first pane here is what? The editor pane. So this is where you will be writing your code. This is where you'll be writing your code. So that's the editor pane. This is document. You'll be documenting your code. This is everything. You'll be doing it here. That's the editor pane. So the next pane we have here is the environment pane. The environment pane here is where the variables that you are assigning, your data, you'll be saying where your data, not your file, your data is saving to, your data that you are assigning variable to will be saving to. And also you'll be seeing histories of what you are doing. So this is what I've been doing in the past before. Then let's say you want a connections, don't bother about this. Don't know if you are familiar with version control, GitHub. You can do it to, push some of what you have been working on to GitHub environment, and this is just tutorials to learn more about Arc Studios. Anyway, yeah. So this is the environment pane. Environment pane as name, the first pane that you are seeing here. This is the editor pane, and then to the console pane. The console pane is where your code that you want to run is going to be working. Because if I said print hello world, It's going to print hello world. This is where the code will be working, but this is where you will be editing the code. You'll be writing the code and then running it here. It's not a bad idea if you want to be doing everything here, but this doesn't look interactive. This is basically what R is made up of. This R, this is what you'll be seeing in only R. If you open R, this is only what you'll be seeing. All right. Let me quickly come back again. I'm very sure that we have the QMD file here. So this is the editor pane. This is where you'll be editing your content, you'll be writing your code, you'll be doing everything like most of the time. But that's the editor pane. It has a lot of functionality, and I'm going to explain all these things that you are seeing here. Don't be scared about what you are seeing here. I'm going to explain some of it, and still, it's not something that you get in a day, to be honest. I'm also learning about some of the I'd say features under development. Most of these things are always under development. You can see features that, but it's because it's still like undergoing development every day, just to meet what user would be interested in using. So that's the editor pane. So this is the environment pane. The environment is empty now because I didn't have anything there, and I'm still going to show you more when I have some code working here. And then this is the history. So all the codes that, so if you want to make connection, maybe you want to make a form of connection with database or Spark or some other connections, you can do where to do connection here. So then Git, I said the version control system. Let's say you are done working here, or for example, let me just use something very simple. This is deviating from the class. Like I said, we do have a 3 month programs that do cover for a lot of explanation about some of these things, which I will still explain later on. This Git is the version control system. Let's say you have a document, like you are working on a project, you name it project 1. So then your supervisor corrected some things, and you don't want to delete the previous project, right? You don't want to delete it, but you want to update new things that you are correcting for what your supervisor said. You are naming that project 2. Then going further, you have a lot of documents in your desktop. But with the help of Git and the remote version control system environment like GitHub, you'll be able to save everything here and save it in the GitHub like this. Let's say my future self, I want to come and see this same thing again. I just need to go to GitHub to pull my information back and then continue to work on it. So that's a lot, but that's just the pretty explanation of what Git is, trying to maintain the version of what you are working on presently. Then we also have tutorials that you can always learn more about RStudio and how to interact with the environment. Now I said we also have the console pane. This is the editor pane. The first one here, the environment pane, then the console pane, and also the plot pane. So now we have the console pane. I said the console pane is where your code will be working, like where your code will be working. Just to execute it, because this is where the R engine is. This is where your code will be working. Like what I just did here, print hello whatever. I say 1 plus 2, going to say 3, because this is where your code is working. I'm still going to show you the interaction between your editor pane and the console pane. That's console pane, and we also have terminal beside it. If you are very familiar with using terminal, which is some of the things we do use in bioinformatics a lot, how to do a lot of things in the bioinformatics. So this is another thing, just to interact in the same environment. So this is terminal. We like Linux based environment, how to do analysis in the Linux environment. Okay, now I think that's great. You've opened the RStudio now and opened the file. That's great. That's wonderful. So now, are you on the same page with us? And if you have any questions, you are lost, please let me know. The reason why we are doing this is because we want you guys to have, if not everything, at least the basic understanding of how to use the R Studio environment and how to do analysis in this environment. All right, so I believe we are on the same page now before moving forward. Now, this is the console pane, right? For R, you don't need to worry about terminal. You don't need to worry about it. In fact, you can delete the present terminal. What we are interested in is what our R console. This is what we are interested in. So we run print hello world, any code that you are running, this is where you will be seeing it. And this is the file. This is where, if you save a particular file, this is where you will see that file. So this is where the file would be. This file, this view, like generate a plot. This is where the plot will save to. If you are developing a package or you want to make use of existing packages, this is where you will see that. If you need help, this is where you see that. If you want to view a document, this is where the viewer will do. If you want to do a presentation, which I will not be doing today anyway, this is where you'll be seeing that. You can do a presentation in our studio environment. In fact, you can prepare a slide in our studio environment. So, and talking about presentation and slide, you can see what I'm using here. I'm using QMD. The file, the extension file I sent to you is what QMD. That's Koto Markdown. Koto Markdown is an open source publishing system. So this is just for us to get, let me open another thing here. This is R. Let's say I'm using just ordinary R extension. This is how I'll be writing my code. This is how I'll be writing my code. This is how I'll be writing the code. Print hello world. This is a test for R language, something like that. My copilot is working here anyway. So print this is how I'll be writing it in .R. But it's rare that you see anybody using this R Studio, and that same person will still be using .R. So that's the only reason why you will still be using R. But for intuitive interactiveness, you would want to use what? Either R Markdown and what, or QMD. We want to use R Markdown. We also have another one called what? R Markdown. We have a lot. I'm just going to click OK. So you can see this R Markdown. You can see this very similar to the Kotlin that I've been showing you, but it's different in the sense that you cannot do a lot of things with R Markdown. Just to keep it short, Kotlin is the next generation of R Markdown. So just to keep it short. So that's that. This KMD, I don't want you to worry about what you are seeing there. This is created while trying to like create this particular KMD file. And what we want to be doing is phylogenetic analysis, right? Phylogenetic analysis. So I can do it to increase the size there, move it backward there. But because what I want to be seeing currently is just this one and what this pane. That's what I want to be seeing. This one and this pane. That's what I want to be seeing currently. So now this is, let me just not assuming, right? If this is your first time in a coding environment, please do well to type in the chat. If you have been coding before, please let me know. If this is your first time, you can do it to say yes or say no. Okay. First time in a coding class, yeah. Okay, first time. Okay. 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 So if this is your first time joining a coding class, don't panic. This is just an environment. Your editor pane, this is the environment pane, this is the console pane, and this is the file upload pane. Okay, so this is R Studio, and the programming that will be running in this R Studio is what? R. Just to say this now, that does not mean the only programming that you can use in R Studio is what? R. So it's still retaining the name R Studio. So R Studio has moved on to what? POSIT, just to say that we are not designing this platform only for R. But though it's majorly for R, but not for R, we still want to allow Python users, we want to allow observable users, we want to allow Julia users and some other programming users, C, C Sharp and the likes, to be able to use this environment. So these are our goals, but we are still developing our ecosystem around R, but we want people in Python, Julia, observable our ecosystems to be able to run the code. So this is this. So let's say I want to, like, this is the QMD, this is the Kotu Markdown, this is the interface for Kotu. You can see here I have my name. While creating the document, it says I should put my name. This, don't worry about this. Am I creating an HTML visuals? This is the visual form. You can change it to source. This is how the source looks like. This is the visual. This is how the source look like. This is the source. This is the source. You can see the differences. It doesn't look more visual, but this is the visual. Most people want to work in this visual, right? I'm going to shift this to this side so that we can see that clearly. So this is phylogenetic analysis tutorial. What we'll be using is what? Hemoglobin genes. Dwelling much into the functional aspect of the case study, but the structure. So that's that. And if you, this is the document. There's two things in the document. If you work with Python before, and you've worked with Jupyter Notebook or Jupyter Lab before, you do have a section whereby you can write documents, you can write content, and you have a section whereby you can write code, right? The same thing is applicable here in the RStudio or Kotu Markdown QMD environment. The same thing is applicable here. About what we want to do today, phylogenetic analysis tutorial, and we are using what? Hemoglobin genes. So that's that. And this case study introduction, hemoglobin is what? Is the oxygen transport protein in red blood cell, and it's composed of what? 2 alpha and 2 beta subunits. The globin gene family arose by gene duplications, so that different subunits evolve in vertebrates. So that's gene duplications. I'm not going to dwell in depth into what this paper is talking about. So I'm not going to dwell in depth into what that paper is talking about, but I just want to show you some other things and how species. For example, alpha and beta globin gene diverged millions of years ago, and various species have their own versions, right? So by comparing the nucleotide sequence of the hemoglobin beta subunit gene across species, we can reconstruct evolutionary relationship among those species gene. So like we want to compare different species using hemoglobin beta subunits. We want to see if there's evolutionary relationship between these species, and then that's basically what we want to achieve in this call, right? So, and there are some other aspects which this paper do explain about, but on the functional aspect of phylogenetics. So phylogenetic trees of hemoglobin beta subunit gene can reveal both speciation and what gene duplication histories. But what we want to focus on is what? Speciation. So I can do what to help you to bold this. The same way that you bold your document. Just see this environment as something that you do in your document. So see it as something that you do in your Word document, right? If you want to put slant, but just like your contour like that. So that's what we want to focus today. And the hemoglobin beta chain is encoded in what? Hemoglobin gene. I want to believe that some of us here, majority of us here rather, we have a background in life science. And me saying something about gene and maybe nucleotide will not be strange to us. If not, please help me to debunk that before moving further. If you are coming from life science background and familiar with what gene is, what nucleotide is, or if you've heard about that before and you are familiar with what I'm saying now, that's okay. If not, please let me know. If you are coming from other background, let's say engineering background to be specific, or maybe pure background on sciences and not biology. We want to assume that we are all coming from life science background. So that means gene, me saying gene, me talking about hemoglobin, beta chain is not new to us. But I'll just quickly talk about what hemoglobin. Hemoglobin is the carrier of oxygen in the red blood cells, basically. So, and we have two alpha chain and two beta chain. Based on evolutionary relationship, it has been reported that most hemoglobin from different organisms, even within the same organism, which is alpha and beta chain, evolves over times, and that's why we're having alpha and beta chain just to meet the functionality demand at that time. Following Lamarck's theorem that says that a characteristics we develop based on demand, like Z giraffe trying to plug apple, right? So snake, that's where that morphology characteristics was developed from. That's Lamarck theories. But we are not going to that, but this is just saying that the reason why we have alpha beta chain is because there is that time there is need for an oxygen carrier, which is hemoglobin, to exist. That's why the evolution occur for that particular gene. So what we want to focus on this call is that speciation. That means different organisms, though they do have they have hemoglobins, well, with different changes in nucleotide sequence, but are they related to each other from evolutionary standpoint? So that's what we want to discuss today. Okay, so we have genes. Gene is coding for protein, right? We have the beta chain that is encoded in the hemoglobin beta chain gene. Sequence differences accumulate over time. So alignment of ABN across species reflect their divergence. So the goal here is what? We want to download. The first thing we want to do is what? We want to download hemoglobin beta gene sequence. If I say gene sequence, I'm talking about nucleotide sequence. I'm not talking about protein sequence. For human, we want to download for chimpanzee, we want to download for mouse, we want to download for cow, pig, and some others. And then after downloading those sequences, we want to align those sequences to each other. And then we want to infer phylogenetic relationship between those sequences. So that's what we want to do in this section. To quickly at time, we will be able to end by 2. So first thing is what? Setting up your R environment. So as before, we need the same package as any new one. Let me be sure if I'm using, oh, this is not the, this is not the coming. coming. So I want to believe we all have our Arc Studio opened. So I think there's a mess up here. I'm quickly going to adjust that. Just going to quickly adjust that so that it doesn't look confusing to us. There's this file, not this particular one. All right, I'm coming. So this is what happens sometimes. I think this particular files I'm looking for, don't want us to stay long with the call. It's not the same file with this one. So not the same file, this one. Anyway, so the first thing is retrieving the sequence, but before then, you want to install packages to the environment. So that's the first thing to do. So you want to install your packages. Install dot package. So we have if we have enter is from NCBI. So but I'll develop a package, like people develop a package for our users. That's our interest. Ape is for phylogenetic tree plotting. We have biostrings to just allow us to read our DNA molecule. Then we have what MSA, we have GG3 for visualization of our plots. So ape will allow us to do the phylogenetic, but GG3 will allow us to do visualization of the plots. So that after having this, what you need to do is what? To run this. This will run your code. You run this, pressing this button. You run that. So that will be running here. Like I said, this is where your code will be running. So that will be working here. So it's still working. You will see this like this colored, red colored, showing that this is working. So it's done from my end. So now moving to Bioinformatics MSc. So why this is running, let me quickly explain the difference. You are installing what? Packages. Packages in R. Packages in R is what? A collection of functions and maybe data as well. But when you are in Python, you call it library. But for here in R, we call it what? Packages. Then I'm installing ape and reinterest. The reason why you are saying install.packages here as what? The function to use is because you are calling that package from CRAN. CRAN is a repository for storing packages. You are calling it from CRAN. So that's the name of the package manager, one of the package manager in R. So that's CRAN, Comprehensive Read R Archive Network. Yes. So we use install package, and I think there's install package. Do about install, but why are we not using ape and then Bioinformatics? Why are we not using install.package to download this package as well? Bioinformatics, MSc, GGT. This is because these packages are not housed in CRAN, whereas they are housed in what? Bioinformatics Manager. For somebody that is coming into the bioinformatics space and into the life science space, like genomic space. So Bioinformatics Manager houses a lot of packages for those kind of users, but this is more of statistics space. But we also use it as a bioinformatician to analyze some of our data. So the reason why you are saying this C here is just telling you that you are concatenating two things together. You're having the RNTS as vectors, you're having the what ape as another one. So you are downloading two things. That's why you are seeing C here. Adding being there's no, you are downloading only one thing. It's just going to be in this format. Install dot package tidyverse. Okay. Tidyverse. So that's how it's going to be. But because you are downloading multiple packages together, that's why you have to put this C there. So that's the first thing. Yes, saying, did I want to install everything? Yes. I'll just say yes. All. Right. Use some or you don't want to install anything. I'll just say all. And if you are using, what's it called? Posit Cloud, I think I still have to resume this. I think you just the same process. So just the same process. You just install it. Same process. You do run this here. It's going to be working. But since I'm doing this in my local environment, I think I should still stick with this. this keeps showing me this. So my code is still running. Well, I'm not sure why this is pretty slow, but that should be working in your, you see something similar like that from your end. Still loading. Still loading. You can see that it's still loading. This takes a lot of time. I should have just stopped it and done. It will work because I've already installed the packages before. I just want to see you guys to see that it's still going to load and then it's going to complete later on. So now that I'm done downloading this, you should have something like the downloader accessor in these download packages. So the next thing I want to do is what, if you're in Python, you need to import that library, right? For R, you need to import it this way, library, then you call the name of the package here, ape, the interest, bow string, MSA. I'll just run it like that. So it's bringing out a lot of things. Just to be sure that it works perfectly fine, I'm going to rerun it again. You can see that it worked perfectly fine. Library works, ape work, the interest work, bow strings works, MSA work, and GGT works. So let me see if I can move to my bioinformatics folder. I'm coming. I want to be sure of something here. Okay, so I think this sequence, I want to edit something from here because I thought I saved a file. So you can see that I'm trying to look for something. So I after running the library, after saving that, after running the library, and you see that all these packages that we want to use is working. So the next thing to do is what we want to retrieve the hemoglobin genes, and you can see that trying to look for a sequence, like I'm trying to look for a markdown rather, but I'm not seeing it currently. Even just to say this, for some of us that are saying that we are confused, it's very common, even to the expertise. So I'm still going to figure that out later on, but just to stick with what I have in ground for you guys, so I'll just quickly edit this. What I want is what I want. I've already worked on NCBI to download to check for accession number of hemoglobin beta subunit from human, from some other organism, chimpanzee, and some others. So what I'll just do here is what I'll copy this. Okay, I'll copy this. This for human. I think that one is right. Then I'll come to this for Pantoglosside should be here. So I'll just copy for this. Then I'll do the same for the next one, which is what? This chlorosabios is this is the best thing I'm going to do at this point. I'll just copy this. Then I'll move to most musculus, which is this, I think. Right. Check, I still have something here to adjust. So I didn't tell you about comments. If you want to comment in now, just the same thing if you are familiar with Python. So you use the comments, the hash sign to comment. So then I'll move to the next one. I think I'm just going to follow this format for that. This is for that. That should be okay. Basically, this is for that. I will just switch this one. No. I'll just come down here to put for that. And I will put it in a string format. Then I would copy this up and then this. So I think I'll put comma here. So you can see that the arrow is pointing here, there's arrow here. So I'll just put comma here. Then what I'll do next is which one is posterior? Posterior should be cow, right? Yeah, it should be cow. I'll change this and cover this up, and then did I really change that cow? Okay, I think it should be here. Okay, then what's next? Source sarcopha, and then source sarcopha, and then Canis lupus. Canis should be. Do you have any canis here? Yeah, we have canis there. Yeah, what else? Where this cattles? Just going to change this to. I'm not sure where I got it wrong. So I think the last one I'm going to change it to equals calibers. For calibers, and then I'll comment this one out. Oh, no, I'm not commenting everything. I'll just comment this one out. So that's two. But I think I'll just need to run this first. Let me works. Now my ID is saved. So now I want to run a faster file and I want to save it as what? Hemoglobin genes. Let me just rename it. Hemoglobin gene dot faster. So I've assigned the ID for this accession number. Accession number is what you will see for most of the sequences in the database. Almost happens, all these are accession number, right? I've assigned it to IDs. These are the IDs we have for these 10 accession numbers. Then the next thing I'll do is what I would fetch it. I'll download those data. What we have here, something similar that we have here. I'll download it from the database. So I'll download it from the database. Don't worry about that. We'll share the script with you later on, because I've modified the script. So I'll run this. You can see that it's working. You can see that everything works fine. So you are expected to see like 100 characters here, because after fetching the sequence, it's going to save in this format, HB faster. Then I'll write that sequence to what? HB faster. Then the name of the file to be what? Oh, sorry. I think I didn't like the name I named the file. So I just need to go back and rerun it again. So I write it like this, hemoglobin gene dot faster, and then I want to cut the content of what? The HBB faster, if it truly has the content of the sequence, and also if it has saved. If it has saved, I should see it here. Let me see my working directory here. My working directory is HP then documents. So I can go to HP documents to see if my hemoglobin gene has been saved. Sure. So maybe I need to set my working directory so that I can have the documents. I'm coming. So I think these are some of the things. Coding is never straightforward, right? So looking for bioinformatics analysis that desktop, desktop first, the bioinformatics analysis. No about there. Yeah, this set working directory. Which is not complete. The reason why you are seeing this plus sign here is because I didn't put this here at the string signs at the end here. So I'll just write something and close it. Yeah. Always difficult to not killing the process. Yes, I'll clear the environment first and see. The process. Tested. I guess I'll have to close this and then reopen it again. Not sure why this is not killing the process. Maybe my system is on. So I'm just going to quickly run through it so that I will not. I'll run this again. Then I would set the working directory so that we can have it in. I've set the working directory here, so come. Yeah, so this is where I want it to be. So now I need to run this again so that I can see the file in question. Hemoglobin again. Yeah, this, then let me run. Okay, I've run this. Let me run this now. So now you can see telling us that this hemoglobin. If I should come to this place, you can see that I'm seeing hemoglobin gene dot faster. So I'm seeing it here, hemoglobin gene dot faster. So that's the faster sequence downloaded from NCBI using R. So that's the first thing we did. This is the faster sequence. For every faster sequence, I know this is very new to you, and this may look a lot. So this is the greater than sign showing that it's a faster sequence. This is the first sequence for human. This is the second for most musculars, and then for others. So, and then I would, after downloading the sequence, the next thing I want to do is what I want to this way. I need to read it into into HBB sequence. In the HBB sequence now I need to set the species, but I don't need this. I didn't label it. The label I'm intending to use, I think I just have to start labeling it the name of the organism again. So I'll just leave the labeling out. So the labeling is just meant for us to know that this is human, this is, I'm still going to come back to that if we have more time later on. So after downloading the sequence, next thing to do is what? Basic next thing to do is what? Multiple sequence alignment. Then you run the multiple sequence alignment on this sequence that you have here. You run multiple sequence alignment on this sequence using what? MSA function. Then you pass the name of the sequence, like the name you assign to that sequence. And the method we want to use is what? Cluster omega. So we have a lot of methods to use. So I'll just run this. You can see using GoNet. So it works and it's showing us the alignment. So it's telling us that this is pretty much where everything aligns. So it's a lot of column. It's not displaying everything for us here. We'll have seen where some of these things align and where there is gaps in these things. So this is how to do multiple sequence alignment. So you just trying to do phylogenetic trace, you want to compare the sequence, right? And that's that. So I will. So that's that we basically see that the first few line, and if you want to see that this is name of the sequence for the first one, and for the second one you are seeing sarcopha as well, sarcopha and Felix catus. like first information about it, which is pretty not necessary for now. So then after doing the alignment is meant for you to see if there's gap, if there's insertion, if there's deletions between these two different sequences. Then you want to do what? You want to plot a phylogenetic tree. You want to convert the alignment to what? DNA bin. So DNA bin is just the object format to save a phylogenetic tree. So that's the first thing. You convert to DNA bin, then you run what? The phylogenetic tree, just to show you what? That a tree is being plotted. So, and let me see, so, and this we do neighbor joining on the distance matrix. It's a lot of statistics which I will not be able to explain in this short call. Then you run the tree and it's going to work. So it's going to print out this information for you. Phylogenetic tree with 10 T's and 8 internal works. And it's telling you the phylogenetic that it does. And then after that, I will run the phylogenetic tree on this sequence. I know it's and it's going to plot the tree for me. You can see here, the reason why I didn't use the label is because it's going to take a lot of time. And I will have renamed this. I will have renamed it here. At this point that I comment on this code here, I will have renamed it and come down here and say species. Since I know that my first species is what? Okay, where is the line that I'm working on? So my first species is coming. I hope we can see my first species is this. So I will just say the first one is what? Human. The second one is the second one should be I should check the sequence here. Second one should be pan-troglodyte. I didn't know the particular organism that is for now. So I'll just say, I'll just come here and say human, or maybe I just want to stay with this naming system. Moss. Let me just name it so that you can see what I'm pointing at. So moss. But before this, I want to show you something from the phylogenetic tree so that you can see the relationship. This is Homo sapien, right? And this is Chlorius sabius. If I should check Chlorius sabius, what Chlorius sabius is, I think I already did that, but I don't know how the file got makes sense. I think it's one Chlorius sabius. Chlorius sabius. I'm coming. I think Chlorius sabius is monkey, right? So, and I'm pretty sure that all these other ones as well that I ran, this equals, pan-troglodyte. Let me check that one too as well, because I want to explain something around that. You can see that that is chimpanzee, right? So, and you can look at the tree. Though the file that I already set for this, I'm not sure where the file is right now, so I'm still going to look into that later on. But this is Chlorius sabius, which is monkey, and then this is Homo sapien, right? Then this is chimpanzee. You can look at it and see that these are very closer to each other. The monkey, these and these are very closer to each other. Moving further, you can also see that of rats. mouse, they are very closer to each other. So that means their hemoglobin genes are very closer to each other from evolutionary standpoint. And you can also see that of human and then the chimpanzee and the monkey very closer to each other. You can also see the cat and others. I think it's just showing that they are closer to each other evolutionarily. So this, I will have renamed it. I didn't been have renamed it here. You will have been seeing it at the front. Instead of seeing all these long labels, you will be seeing it as either man, chimpanzee, and the likes, which is what I'm trying to achieve in this particular line of code. Line, maybe the third one is going to be chimpanzee. Something like, is it dog? I'm not sure for now because I've messed up the naming. So continue to 10 like that. So that's what I'm trying to achieve. So that so whenever we want to check this, like we want to check the phylogenetic tree, we will be saying instead of pantoglo that here, you'll be seeing chimpanzee, and for this you'll be seeing monkey. For this, I think it's cats and some others. Instead of seeing all these long names, so just for you to get that this is there's a relationship between this. So that's that, and so that's that. I don't think I'll be able to go through these questions, and I think that's all I have for now. So, do you have any questions before? Any questions? Why is my screen stopping here? Okay. Do you have any questions?
If you have any question, you can type it down to the comment section, or you can raise your hand with an emoji.
If you have any questions, if not, I think I still have some other things to share. Yeah, sure. The recordings will be shared. I think it's going to be via YouTube channel anyway. That will be shared with us. And just before then, I would like to, if you are more interested in what we did today, I think what we did today is very pretty. It's for somebody that is just coming into the bioinformatics space is very pretty, like bulky, right? But we do have a training that spans the period of 3 months. So if you are interested in registering for the training, I think I will just share the link with us in the chat so that we can. I think I do have something that we do bioinformatics related stuff around healthcare. I'm still going to explain more on that. Let me just share this with us in the chat. So you can do it to check that, and so for that so if maybe you are professionals in this space already and you are looking forward to maybe becoming somebody that can analyze data and epidemiologist, can do it to look at our training here. So, and we have a lot of curriculums around Earth data analysis, visualization, SQL, and some other case studies to work on projects, then predictive models and data quality assessments, building machine learning models, doing evaluations, working on medical imaging using CNN, then bioinformatics aspect as well, especially around uh, protein structure predictions and doing some other cool stuffs in bioinformatics. If we do have any questions, please do it to unmute yourself or type in the chat. Any questions? What I just said today is pretty much introduction, how to download on the data and then plotting phylogenetic trees. I know the coding may look scary, so and not expecting you to get it at once, but anyway, if you have the recording at the end of the day, it will, I believe it's going to strengthen what you can do with the bioinformatics, like the phylogenetic analysis. Yeah. Any other questions?